up. It's my pleasure today to welcome Ruth Wilkins to give us our Friday noon talk. So Ruth is the Division Chief of the Ionizing Radiation Health Sciences Division at the Consumer and Clinical Radiation Protection Bureau of Health Canada and has been in this position since 2006. Ruth is also an adjunct professor in the physics department at Carleton University in Ottawa and is a lecturer of graduate level radiobiology course in the medical physics program at Carleton. And Ruth got her Ph.D. from Carleton, has a MSc in biophysics from UBC and a BSc in physics from Trent University. And Ruth is an expert on biodosimetry and the biological effects of ionizing radiation. And today she's going to give us a presentation on biodosimetry at Health Canada. So over to you, Ruth. Thank you very much for joining us.
Well, thank you. Thank you for inviting me to speak at your lunchtime seminar. And I hope that today I will teach you a little bit about biodosimetry and talk about the biodosimetry program at Canada and also how it's a very much an international activity. So I'll just give you a brief outline. I'll tell you a little bit about Health Canada, where I come from, what is this CCRPB we talk about, then talk about an introduction to biodosimetry in case some of you have no clue what biodosimetry is. And then I will talk quite a lot about biodosimetry for emergency response and then just some concluding remarks. So to start, Consumer and Clinical Radiation Protection Bureau, it's quite a mouthful, CCRPB as we call it. We are part of Health Canada and we work on everything non-nuclear, including noise. So any radiation that isn't from a nuclear source but also acoustic. So the whole spectrum from non-ionizing to ionizing radiation. Our mandate is to manage the risks from these types of radiation and through this, we administer the Radiation Emitting Devices Act. This is our beautiful building in Ottawa, which is currently surrounded by picketers, unfortunately. CCRPB, our organization chart, we have a few units, the business standard units, there's a risk management group, but down here we have the Ionizing Health Radiation Sciences, which is me. And my group, and then we have non-ionizing radiation health sciences, and then we have two kind of parallel physical sciences divisions for ionizing and non-ionizing radiation. Within my division, we really have two activities. One is providing biodosimetry, which is the topic of the talk. I'll talk a lot about that, and also just looking at the low dose radiation effects of radiation, looking at different biomarkers of effects of low dose and trying to understand what's really going on at the low dose area of the spectrum. And this is all within ionizing radiation. So just our mandate in our division is to provide biologically based dose estimates for occupational and accidental exposures for ionizing radiation. We support the Federal Nuclear Emergency Division, the Canadian Nuclear Safety Commission, and the Canadian Space Agency. We also conduct research on biological effects of ionizing radiation, including identifying novel biomarkers and the development of new biodosimetry methods. And we are trying to improve the understanding of the effects of low dose of ionizing radiation. So our key deliverables are to maintain a robust and reliable biological dosimetry capabilities for Canadians and for emergency response, and to continue to develop a strong low-dose radiation research program and coordinate low-dose radiation nationally. So that's sort of all the bureaucratic, boring stuff out of the way. So let's get into the more interesting part of the talk. I'm going to give you a background on biodosimetry to bring everybody up to speed on what biodosimetry is, and then get into some more details. So what is biodosimetry? It's very simply, it's a method of measuring a dose received from ionizing radiation using biological material from the exposed individuals. So this can be used in the case of an accidental exposure, or sometimes a physical dosimeter may have a dose registered on it, but the person wearing that dosimeter does not think that they received that dose, so we can determine whether or not the individual actually received the dose on the dosimeter. Sometimes the dosimeters aren't even available in cases where someone's exposed, but they're not a nuclear energy worker and not wearing a physical dosimeter. It can be used before and after time spent in space, because astronauts get exposed to higher levels of radiation than we do on Earth. And it can be used in the case of a radiological terrorist attack or a dirty bomb or any other large-scale event involving lots of individuals. So we provide biodosimetry for occupational exposures for the Canadian Nuclear Safety Commission. As I said, we support the Federal Nuclear Emergency Response and the Canadian Space Agency. So how can biodosimetry help? So for occupational exposures, we're really just looking to determine whether that individual has exceeded their limits of radiation exposure, so that if they have received too much radiation, then their job could be altered for the rest of the year to minimize the exposure to radiation occupationally. But for large accident, I should say, so in this case, for occupational exposure, we're really trying to measure fairly small doses of radiation below 100 millisieverts. So we need to have a very accurate method for biodosimetry. But for large exposures, when we have large accidental exposures, in that case, we're really looking to determine if those individuals need any medical intervention to help them get through any acute radiation syndrome or any adverse outcomes from the radiation exposure. So you can see on this graph here on the right, this is just an LD50 graph, so a lethal damage graph, which shows you that as you increase the dose of radiation, your risk of death will increase with this sigmoidal, very typical shaped LD50 curve. And from this, you can see that the LD50 from this graph, which isn't really quite accurate, but the LD50 for humans here is about around 3 gray. That's probably a bit low. So this would be what your LD50 curve would look like if you had no medical intervention. So you can see at about 3 gray, about half of the individuals would die from the radiation exposure. If you can identify these people, then you can provide appropriate medical care that can help push this curve towards the right. So you can, just by giving some minimum supportive care, like some antibiotic treatment or some cytokines, you can shift this curve a little bit to the right. And if for higher doses, you can provide more intensive care like stem cell transplants, which can really improve the outcome of these individuals. So someone who, say someone received an 8 gray acute whole body dose of radiation, without any medical intervention, they would die. But if you give them a stem cell transplant, then you greatly improve their chance of survival. So it's important to know what dose these people have received, because you don't want to be wasting resources on those that don't need it. So you don't want to give people who have received low doses intensive care. You also don't want to waste resources on people who have very high doses, because they will die no matter what intervention is provided. So it just allows you to identify those individuals that will have the most benefit from the medical intervention and what intervention they should be provided. So how do we do biological dosimetry? There are many methods. I'm just going to talk about 4 of them today. There's a Dicentric assay, the cytokinesis block micronucleus assay, translocation analysis, and gamma H2AX. And these are all based on damage to the DNA, and we measure them in whole blood samples. So just to give you a little bit of background on radiation damage to chromosomes, for those who aren't familiar, you can really classify radiation damage to chromosomes into 2 main categories of unstable damage and stable damage. Unstable damage is damage that is lethal to the cell. So there are gross chromosome changes, and it renders the cell unable to pass through mitosis, and they disappear with time. So an example of this is the Dicentric chromosome. So this is a metaphase spread. So all of the chromosomes are in the most condensed form. So you can see the individual chromosomes, and you can see that each chromosome has 1 centromere. So 1 point where the 2 chromatids are attached. And if when the blood is irradiated, these chromosomes can be broken. So if you have 2 chromosomes broken, they can repair with 2 of the chromosomes with centromeres joining together and 2 pieces without centromeres joining together. And then when this chromosome goes through replication, it forms this very characteristic dicentric chromosome that you can see here on this picture. So here's a dicentric chromosome and an acentric fragment that accompanies it. So this is a nice measure of radiation damage. Another type of damage from very much similar exposure, where you have two chromosomes being cut in the same way, instead of the two pieces with centromeres coming together, these two pieces could just switch places and you end up with two chromosomes that look morphologically similar, but they are in fact translocated. So these are considered stable. They're not lethal to the cell. The damage does not stop the cells from dividing, so they can pass into mitosis and into daughter cells. And you can see, I will talk a bit more about this. You can see some of this by some staining that allows you to see where these chromosomes have translocated. So a little bit more on the Dicentric assay, because the Dicentric assay is the gold standard for biological dosimetry. It has been around since the 1960s. And with this, we just count the number of unstable chromosomes in a sample of blood from an individual who's been exposed. It's very sensitive and specific to radiation. If there's a low background of Dicentrics, if you count 1,000 cells, then a typical person would have one, maybe two, but mostly like 0 to 1 Dicentrics per 1,000 cells. So that'll gives you a very low background over which to measure the damage. And the sensitivity of the assay is about 0.1 gray when 1,000 cells are evaluated. So here we have again the Dicentrics, and this is just a normal metaphase spread. As I said, this is unstable damage, and the damage decays post-exposure with a half-life of about 3.5 years. So it's best suited for acute and recent exposures. So what we do is we generate these calibration curves or dose response curves for radiation. So we take blood from an individual who has not been irradiated. We irradiate their blood, we count their damage in their blood, and we can generate this calibration curve, which is as a function of dose. And here we have Dicentrics and rings, but mostly we just count Dicentrics now. And you can see that for low LET radiation, you get a linear quadratic shape. And for high LET radiation, there's a linear dose response. And then if we get as an unknown sample, we can measure the damage in that blood sample and then just read off the graph what dose that individual received. So the cytokinesis block micronucleus assay is another assay that we use that also identifies chromosome damage. It's a little bit less specific and less sensitive to ionizing radiation. It can be caused by other types of stressors. So in this assay, it's very similar. We're measuring chromosome damage. So you can see here that when a cell gets irradiated, you can get some broken chromosomes that lose the ability to be incorporated into the daughter nuclei when the cell attempts to divide. So it's lost, like these fragments cannot be incorporated into the daughter nuclei, and they end up just forming their own little micronuclei in the cytoplasm that forms its own nuclear membrane. So you can see in these cells that if we stop them just before the cell divides, but after the nucleus is divided, you can see these nice binucleated cells and just count the number of micronuclei within the cytoplasm. And similarly, we can generate a dose response curve as we did for the Dicentric assay. And then translocation analysis by fluorescence in situ hybridization. This is the method that we use to measure stable translocations, which are long lasting, so they're good for chronic low levels of exposures. Again, they're not as specific to radiation as Dicentrics. They have a lower limit of detection of 0.1 to 0.2 gray, but only if a pre-exposure sample is acquired. So it's quite well suited for astronauts, and this is where our work with the CSA comes in, that we can get a blood sample from the astronaut before they go into space, and then we can get a sample when they come back and compare their translocation, increase in translocations while they're in space, and use that as a measure of radiation exposure. So here are just some other images of some translocations. You can see this, these two chromosomes have translocated, and here's another example over here of a translocation. And then the final assay I'm going to talk about is phosphorylation of H2AX, which we call gamma H2AX. H2AX is a variant of the H2A histone family, so it's a histone that is found in the DNA, and it's rapidly phosphorylated upon the induction of DNA double-strand breaks. So when the DNA gets damaged, the H2A is phosphorylated to H2AX, and it's actually recruits DNA repair proteins. It's a signaling factor, so it recruits the DNA repair proteins to the site of DNA damage. And you get one of these foci, if you can see on this slide here, these little dots are the foci that are formed when they're fluorescently labeled. You can measure them as a function of dose. So this is in macaque monkeys, and in humans, you can see a linear dose response curve, which goes actually much higher than 2 gray. You can also, the one problem with this assay is that as the DNA double strand breaks become repaired, then the H2AX becomes dephosphorylated and the signal disappears. So you can see quite a rapid decrease in the signal as a function of time. So you have a very small window of opportunity to measure the damage with this assay. So I like to say that this isn't really a biodosimeter as much as it is an indicator of exposure. So if you get a sample that has some foci in it, there's a good chance that sample has, that person has been exposed to radiation. But because of the rapid kinetics of the signal, it's very difficult to determine exactly what that dose is. But it's a very good triage tool because you could quickly identify which individuals have been exposed and then use one of the more complicated assays of the chromosome assays to do a more accurate dose estimate. So that kind of covers all of the main methods that we use in our lab anyway. And so I'd like to now talk about how this would be used in emergency response. So for emergency response, what I didn't mention with the cytogenetic methods is that they're very time consuming. So the first thing I showed you that those chromosomes have to be in metaphase for you to be able to measure the damage. And to do that, you have to culture the blood for 48 hours to get the cells into metaphase. So initially you have 2 days of just waiting before you can actually perform the assay. And then once you've done the assay and made the slides and scored them, as I said, for low doses, we score 1,000 cells, which can be quite time consuming. So we need to find a way to improve throughput so that we can deal with mass casualty situations and get biodosimetry dose estimates back to the medical community in an appropriate amount of time. So we've had a few strategies for increasing throughput for emergency response. And these include standardization and harmonization, improvements in methodology, networking and interlaboratory comparisons, and automation of methodology. And I'll go through each of these one by one. So to start with, standardization and harmonization, these are key for robust performance. So we're really working towards having international harmonization, so we'll be able to share samples, and that I'll talk about more within the networking. So in order to do that, one of the things we've done is we've written standards for the International Organization of Standardization. And then there's an IAE document that has been written. This is the 3rd version of it, which is kind of the Bible of the best way to do biodosimetry. And then also through interlaboratory comparisons, we can improve our standardization between labs. This is just a list. I won't go through in detail of all the standards that our working group, the biological and physical retrospective dosimetry. These are all the standards that we have published over the years. So one thing that we can do, another thing that we can do after standardization is just do some simple improvements to the method to improve the throughput. And this is one very simple thing that we did at Health Canada that made quite a large change in the throughput for the assay. So when I started scoring chromosomes many years ago, and the traditional way to score them is to ensure that every metaphase that you score is a complete metaphase. So that means that you need to count to make sure that you have 46 chromosomes in each set. So this is quite time consuming. And the other thing we do, as I mentioned, for occupational exposures is that we score 1,000 cells because we want to have a very low sensitivity to identify those low doses. But for mass casualty situations, we don't need that same level of sensitivity. We only need to know if someone needs medical intervention. So knowing if they've gotten above or below 2 gray is really the sensitivity that we're looking for. So we can immediately decrease the number of spreads that we score to 50. And then we can also after many years of scoring, you get quite good at determining, just by looking quickly, that a spread is pretty much complete. Whether or not it has exactly 46 chromosomes or not is probably not important. So we developed this quick scoring method that would remove that time-consuming aspect of the Dicentric assay by taking away the counting of the 46 chromosomes, and instead just having a quick look and saying, does this cell look damaged? So if I look at this cell, for instance, I see quite quickly two acentric fragments and this dicentric. So I can say with some confidence, then I could take a quick look around and say, I don't see any other damage. Let's move on and score the next cell, instead of spending a lot of time counting how many chromosomes are in each cell. So this really increased the throughput just by, it took, instead of taking about a minute to score each cell, for normal cells, I can now score them in seconds. And even a damaged cell can maybe take me 20 seconds just to be sure that there's no other damage in that cell. So we did a validation of this method with AECL, which is now CNL. And we just generated dose response curves using both the Dicentric quick scan and the conventional methods for scoring. This is the data from CNL. This is our Health Canada data. And these two curves were found to be statistically similar, or not statistically different. So then we started using the quick scan in our interlaboratory comparisons. So that's just one example of how you can improve throughput. I'm going to come back to some other examples when I talk about automation later, but now I'd like to move on to talking a little bit more about networking. So there really is a need for network. So there are many labs. I'll show you in a moment a graph of all the labs around the world. But for us to be able to deal with mass casualty situations, it's important that we all work together and we all use the same methodology and we're all standardized. So we've started to form these networks around the world so that we can share the workload. And this, as I said, it requires protocols, interlaboratory comparisons. So you need to verify that the dose estimates from all the partners within your network are going to be the same. They also have some added value. The networking has allowed more harmonization to occur. Networks now perform interlaboratory comparisons internationally to expand their networking to an international level. And the expertise across these laboratories can be leveraged for other methods development so we can improve the field of biodosimetry. So let me talk about our network, which I call the rise and the fall of the Canadian Biodosimetry Network, as you will see. So prior to 2002, Health Canada was the only lab in Canada that was performing biodosimetry for the occupational exposures and emergency response. In 2002, we developed a network. We started to develop our Canadian network. So we incorporated 3 other labs that were full biological labs, had cytogenetic experience, and they had their own calibration curves. These were called our reference labs. So these are the labs with a lot of experience who could do dose estimates. So we had DRDC, which is a National Defense Lab, McMaster University, and at the time it was AECL. Then we started recruiting cytogenetic labs from across the country as satellite scoring labs. So we went to one of their conferences. We taught them how to score the Dicentric assay, which was very easy because we're all cytogeneticists. We sent them home with slides. They sent us back their scores, and we found that they were able to do the dosimetry quite easily. Then we started a training course where we would train cytogenetic students. And it turns out that there's only 3 places in Canada that train cytogenetic students. There's the Michener Institute in Toronto. There's the Saint Justine Hospital in Montreal. And then there's a school in British Columbia. So we set up a training program and where we could train cytogeneticists during their training course about biodosimetry. So we we spent one day at each school and taught them how to do biodosimetry. And then these purple stars are just indicating where these students have ended up. So by training it just, we actually didn't train in BC, but by training it just in Toronto and Montreal, we were able to populate cytogeneticists across the country who knew, or at least had heard of biodosimetry. In 2013, the lab at DRDC was sadly shut down. The training program was canceled, and the cytogenetic labs stopped participating in our intercomparison. And then ACL got rebranded. That's not a bad thing. And then finally, the master lab shut down. So that's why I call it the rise and fall, because we are now back to, at least we have increased capacities from what we had before, but now just Health Canada and the Canadian Nuclear Labs perform biodosimetry for the Canadian network. But it's not all bad news. As a biodosimetry network, we started performing annual intercomparisons, and word got out that we were doing these intercomparisons, and many labs started asking if they could join into our intercomparison. So we had originally had a lab at the Armed Forces Radiobiology Institute and one at the Oak Ridge Institute for Science and Education, which is the REACS lab. Then other labs started asking if they could join in. Then international labs started asking if we could join in. So now we've expanded. We have, over the years, all of these labs have participated at least once. And now we have South Korea, Taiwan, Argentina, and 2 labs in Singapore who are participating in our intercomparison. And this is just a table showing our intercomparisons over the years. So typically what an intercomparison comprises of is taking 10 blood samples, irradiating them to a known dose, blinding them, shipping them to each of these labs. So each lab receives 10 samples with the same 10 dose, with 10 different doses, but each lab get the same samples. Then they do their assay of choice and return a dose estimate to us. So you can see the number of labs we've had over the years, anywhere from 4 to 10 labs. And then some labs most of them would perform the Dicentric assay, either the conventional or quick scan. Some would do the CBMN assay. And then this is just the number of scorers that have performed. Each lab would maybe have multiple scorers. So these are the results over the last so many years of the scoring, looking at, this is showing the percentage of samples that were correct within half a gray of the physical dose. The dark blue bars are the conventional Dicentric assay, the gray bars are quick scan, and the light blue bars are CBMN. So when we look at just the Dicentric assay, we get a success rate of about 80%, which has stayed fairly steady over the years. The CBMN assay performs a little less well, which is sort of not that surprising. The other thing we did with these is we timed how long it took to score each of these to show how well the quick scan is really doing. So what I have here is conventional and quick scans with 10 and 50 cells scored. So you can, if we just look at the 50 cells, you can see that it took 60 minutes per sample. So that's scoring 50 cells. On average, took 50 minutes per sample. But for quick scan, that took about 20 minutes per sample, which just is a little bit longer than CBMN. So the advantage of CBMN is that it was quick, but the quick scan is as quick as CBMN and much more accurate. So it's a better method for high throughput biodosimetry. So this is just briefly the experience with our network, but there are many networks around the world, and this map shows the results of a survey that was conducted in 2020 with the WHO that shows all of the biodosimetry labs around the world, each one indicated by a red dot, and the size of the dot is an indication of the capacity of that lab. What this also shows is other networks. So you can see we have our North American network here, if we can include the US. There's a Latin American network, there's an Asian network, and then there's a European network, which I will talk about a little bit more because it's a very active and large network and is doing wonderful things in the world of biodosimetry. So with this European network, they have performed multiple intercomparisons over the years. And, you know, we thought our intercomparison was quite big, but when we had 10 labs in our intercomparison, we thought we were doing really well. But this RENAB, which is Running the European Network of Biological and Retrospective Physical Dosimetry, is what RENAB stands for. They have conducted several intercomparisons over the year, and these are quite large. So they've had labs, this one with 32 labs altogether. So different sizes through the years. They've had RENAB labs participating, but they've also invited labs from outside of their network to participate. So we've been able to participate in their intercomparisons over the years. I'll just focus a little bit on this last one, which was the last intercomparison in 2021 that had over 100 contributors. It had specialized teams, 85 specialized teams, some just for doing slides or some were doing scoring. There were 47 institutions, 27 nations, 3 continents, all taking part in this intercomparison. And they made this nice little logo. And here's Health Canada right here. I won't go into the details of this intercomparison because it would take the rest of my time. But all of this has just been published in a special issue that has all of the results from the many different assays that they had included in their intercomparison. It was quite an undertaking. So I'd like to spend the rest of this talk talking about automated analysis because I think this is really the way for increased throughput. Automated analysis can mean many things. There's automated sample processing with liquid handling and robotics, which I will not talk about today. But there are automated methods for acquiring data and automated methods for image analysis. So I'm going to discuss these last two. So let's talk about data acquisition. So automated scanning and capturing of images from these instruments can be applied to all of the 3 methods that I discussed. So it can remove the time-consuming aspect of scanning slides manually and allows for quick relocation of the cells visualization. So you can take this platform here will hold 8 slides, and then this system here you can stack platforms of slides that can be fed onto the microscope. So you can set it up with, I think, 200 slides and just walk away, and it will scan through all the slides and identify where the cells or metaphases are that you want to identify. So you can see here for the Dicentric assay, it will identify all your metaphases for you. For the CBMN assay, it will find all your binucleated cells. And for the gamma H2AX, it will identify your lymphocytes. And then you can go back to those sites and do the analysis on each of the cells. And this is quite a time-consuming portion of the analysis to do manually. To manually scan through the slide and find each of these metaphases or cells can take quite a lot of time. So this really improves the throughput. So then we can do once these metaphase finders have captured the images, then you can very quickly run through the images and identify the dicentrics. So there is automated dicentric scoring that can be used to then analyze those images. So you can see here's a metaphase spread, and this software has identified 2 of the chromosomes as dicentrics. It puts them up here in the corner so it's easy to see. So when this was first done, it was found that there were a lot of false positives that were detected by the system. There's a lot of times where they're just 2 chromosomes close together or twists in the chromosome. It's difficult for the software to differentiate between these and true dicentrics. So what was quickly decided was that you could mitigate this by quickly running through each of these images and looking at these dicentrics that are placed up in the corner and just agreeing or disagreeing whether these were actually true dicentrics. So you're just verifying whether the dicentrics are true or not. So this was first done at IRSN in Paris, where they found that by doing this verification, they were able to get a fairly nice dose response curve. So this is their automated dose response curve. This is their manual scoring. And what you see right away is that this is a much shallower curve, but it doesn't really matter as long as you are scoring your unknowns with your the same way that you scored your your calibration curve. So if you're always doing manual analysis and you do your manual analysis on your unknown, then you would use this curve. But if you do an automatic analysis on your unknown, then you can use this curve, and it will be a sort of a systematic response, so that you would still get the same dose estimate as long as you match the analysis for each with the curve that you have used. This method has also been verified by another lab, the Elspeth lab, and they have done a fully automated curve here, so no verification with an assisted. So you can see here the difference when you do this verification. You can also see down here at the 0 point that you can decrease the false positives. So this can greatly increase the throughput. This is a very, very quick way of scoring once it is fully validated. You can also do this automatic microscope scoring for the CBMN assay. Same idea, and here's some data from Varga that shows the visual count, so a manual count versus the automatic count, and that you get a fairly linear response. So you get a nice linear increase or correlation between the two. However, if you plot the 1-to-1 ratio line, which would be expected if the automated count was counting as much as the visual, you can see again there's much reduced identification of these micronuclei as you have with the manual scoring. And this has been reproduced by many, many labs. So you can see again here, all the automated scoring curves are much lower than the conventional scoring. But again, it doesn't matter as long as you use the same method for scoring the unknown. And then the same method applied to the gamma H2AX. And in this case, the manual and the auto don't seem to be very different. So here is the dose response curve that you see with manual and automatic scoring after 4 hours after exposure. And as I mentioned, the kinetics of this, the number of foci decrease quickly with time. So after 24 hours, there's still a bit of a dose response curve, but it's much reduced from the 4-hour signal. So as I said, this decrease, the change between the auto and the manual doesn't really matter. And this has been shown through the application of these automated methods to intercomparisons. So if these, this is one of the RENAB intercomparisons where some people scored the Dicentric assay with manual and auto. So if you just look at the blue dots for each of these, and this is the estimated dose for all of these different samples, you can see there's not a big difference between the red and the blue dots, showing that the automated scoring performed just as well as the manual scoring. And similarly for the CBMN assay, another intercomparison, and this is looking at the Z score. So how far away is the dose estimate from the expected, or anything less than 2, or between -2 and 2, Z score is considered acceptable. If you look at the green, which is the semi-automated, and the blue, which is the manual, you'll see, again, not much difference between the two. So showing that even though the dose response curves are less steep than for the automated than the manual scoring, the dose estimates are still as good for emergency response. And so the gamma H2AX has also been tested in intercomparisons, and it was found that at the 4-hour time point, they were able to appropriately categorize the majority of samples, suggesting that the gamma H2AX assay could be used as a triage tool following an acute radiation exposure. So further automation can be done with imaging flow cytometry, and this is the work that we're doing in our lab. So imaging flow cytometry combines the power of traditional flow cytometry with the high resolution of cell imaging. So on the image stream X, which is our flow cytometer, there's a CCD camera that captures the images of every cell. So if you're familiar with flow cytometry, you will know that for each cell that is interrogated by the flow cytometer, you get an electronic signal that will tell you that the signal is on the cell and and how big is the intensity of that signal. But for imaging flow cytometry, you take your cells and you label them fluorescently for whatever marker you're looking for, and it will give you all of the same information of the flow cytometer, but it will actually take a picture of each cell with each channel of the fluorochrome, which you can then overlay. So you can see here, it will do a bright field image and then an image of the cell with all the different fluorochromes that it's labeled with. Then it can take these images and overlay them. So you can see where damage, where your markers are co-located. You can also get traditional flow cytometry plots with dot plots and histograms, providing all sorts of information. So it allows you to do all sorts of image analysis that you can't do with traditional flow cytometry. It's really a very powerful tool. So we have taken this technique, and we have, the first thing we tried to do was adopt the Dicentric analysis to the imaging flow cytometer. So with the Dicentrics, what I showed you are metaphase spreads, and you can't really look at whole cells on the flow cytometer because you won't be able to see the individual Dicentrics. So what we did was take those metaphase cells, blew apart the cell membrane and the nuclear membrane. So we just had a soup of individual chromosomes, essentially. And then we fed these chromosomes one by one into the flow cytometer with a centromere label and a DNA stain. And these are some of the images that we got. So you can see these individual chromosomes, and here are monocentric chromosomes, and here are dicentric chromosomes. And we looked at them as a function of dose, and you can see that they started to increase with dose. Well, you might also notice here that it's not very sensitive. In the low dose region, we would have a hard time even detecting cells that have been exposed to 2 or maybe even 3 gray. So this method still needs a lot of work. Unfortunately, we had to shelve this for a while, but someday we'll get back to it. And I think we need to improve the sample prep for this and to get a better population of chromosomes and better staining. They tend to stick together, which can cause a lot of troubles. If you stick 2 monocentric chromosomes together, they're going to look like a dicentric chromosome. So there's some strategies that we have in mind for trying to improve this assay. But the one that we spent a lot of time on and have done well developing is adapting the CBMN method to the imaging flow cytometer. So the advantage of this is that you can take your cells from your culture, put them right onto the flow cytometer after staining, and you don't have to make slides, which can be a time-consuming step of this assay. The cells can be imaged at over 200 cells per second, and then you have a template that allows you to identify the binucleated cells and the micronuclei. So here you can see this is what our template looks like. We start with a whole bunch of cells, and through a series of gating strategies, we end up with our binucleated cells, and then we can identify how many micronuclei are in each cell. So this software can mask the cells. So we can use masks to identify our binucleated cells. And once we have identified our binucleated cells, we can then use another mask that will identify what the micronuclei are, just by defining the size and the shape of what these spots are that we're looking for. And then the flow cytometer will automatically count how many micronuclei are in each of our binucleated cells. And then we can generate a dose response curve. So typically we only go up to 4 gray, because you can see this has a nice linear quadratic increase. We've tried to push this assay out to the 10 gray part. But what happens at the higher doses is that the cell cycle starts to slow down. The more damaged cells don't go through mitosis. You don't get damaged cells in cytokinesis. So you tend to get a decrease in the number of micronuclei per cell at the high doses. So to use this as a dose response curve would be an issue, because if, say, you had 0.1 micronuclei per cell, you would get 2 answers from this curve. It's either a very low dose or a very high dose. So and this is demonstrated here, where we took blinded samples and we irradiated them and looked at just the lower half of the curve to see if we could identify our doses below 5 gray, and we could do that quite nicely. But then when we went up to the 10 gray level, we saw that this all fell off, and we couldn't really do dose estimates at the highest level. But the advantage of the imaging flow cytometer is that you can use all sorts of other features. You're not just limited to looking at the number of micronuclei and binucleated cells. We could look at how many mononuclear cells or how many quadrinuclear cells, and look at all sorts of different features which we did. We took a bunch of other features and used these features to, with a linear discriminant function, to identify whether the cells were above or below that cutoff of, say, 5 gray, and then do the dose estimate based on which side of the curve it would fall on. And these are the results that we got. So we do very well still in the low dose. These red cells are the red dots are the ones that were correctly identified by the linear discriminant function. So all of the correctly identified ones, we got pretty decent dose estimates for. All these black ones were attributed to the wrong side of the curve. So these samples were not well identified, but it does improve your ability to look at the high dose region. And then finally, I just want to talk about the work we're doing currently on developing the gamma H2X imaging flow cytometer very briefly. This is some work that was first shown by another group at very low doses. So this is at 50 centigrade. You can see the increase in the gamma H2AX on the imaging flow cytometer. And you can use the cytometer to do spot counting on these. So it'll automatically count how many spots are in each cell. And this is data that's just hot off the presses. We've just finished last week, where we had generated a dose response curve in lymphocytes and counted the foci, and we get a fairly nice increase up to 10 gray. It's a little bit of a fall off at high doses, and we're working on our spot counting template to try and improve this linearity. So that is what we are doing at the moment. So just to conclude about automation, we've had a great effort in automating all aspects of the process for biodosimetry. Typically, the dose response curves are less steep, but as long as the same methodology is used for the dose response curve and the unknown sample, the dose estimate should be accurate. So that's just demonstrated here, that for an automated analysis, your unknown sample is going to have a lower frequency than your manual. So you'll still get the same dose estimate. And that was shown through the intercomparisons. So there's definitely an improvement in the time required for analysis, especially with the introduction of cloud-based computing, and also with machine learning and AI. These are now being incorporated into image analysis. So these should also help improve the identification of the damage, which should be able to increase the slope of the curve and decrease the false negatives. I'd just like to conclude by saying the biodosimetry is an important tool for determining the level of exposure for occupational exposures and for individuals in the case of accidents and malicious acts. Standardization and harmonization has been accomplished through ISO, IAEA, and the WHO. Networking is essential for large-scale responses, and the interlaboratory comparisons are key to their success. We have new methods that are constantly being developed and automated that are going to assist with large-scale responses. And we have, through all of this work, developed a strong international biodosimetry community that's been coordinated through our organization that we call the International Association of Biological and EPR Radiation Dosimetry, also known as YABIRD. And you can visit our website here if you're interested. I'd just like to finish by acknowledging all the people that have contributed to this work over the years. And thank you for listening. This is just the group of our YABIRD members and our WHO BioDoseNet people at a meeting in Munich a few years ago, pre-pandemic. So I'm happy to take any questions.